DNA, the music of life. DNA is the blueprint of life because it contains the instructions needed for an organism to grow, develop, survive, and reproduce. My passion is to decode this blueprint and reveal the unique and amazing information hidden in the genomes. Hello everyone, I am Sunil Kumar Sahu representing Beijing Genomic Institute, Shenzhen, China. In today's talk, I will be covering the harnessing the power of genomics in agriculture. So, in today's talk, I will be covering the background about the genomics and various omics technology, and I will be talking about the 10,000 plant genome sequencing project and its application using the microbiome and lettuce as an example, and I'll be also discussing about some of its further applications. So, if we look at the past 2 centuries, we have witnessed a massive increase in the climate change, especially the global temperature. And this climate change is having a significant impact on the agriculture. Climate change causes erratic weather patterns, extreme temperature, and changes in the natural resources, thereby threatening the farmer's ability to sustainably produce and maintain the quality crops. And in the recent years, we have now entered in the era of omics, and the swift rise of omics approaches allows us investigate the microbial diversity and the plant microbiome interaction across different ecological communities and at the spatio-temporal scales. And these multi-omics approaches now inform us from the genotype to the phenotype cascade. And these omics approaches from this, like for example, the genotypes, and we can say it inform about the disease ecology in a holistic manner, and it also can shed light on the microbial communities above, both above and the below ground. And here you can see the red color, it actually symbolizes the dynamic intra-plant communication in the metabolomics approach if you can use, and it can affect the overall microbial community through the release volatiles, and this has led to the emergence of volatilomics. And likewise, we have witnessed that different kind of omics in the recent year, like metagenomics, which I'll be covering in the subsequent slides, and transcriptomics, proteomics, etc. Life is digital, life is information. Now we are trying to bring the digital era formats to omics and especially the agriculture, and heterogeneous omics data are being now generated at unprecedented rate and volume to advance in the biological data acquisition technologies. And this is mainly due to the significant drop in the cost, the wider access to the omics platform, and consequently, in the future, the data storage, transmission, the visualization, and the scalable processing will become highly important for the advancement of both biological as well as the agricultural research. And before moving further, I would like to briefly introduce about my Institute of Digital Earth Bio. It is the main undertaker of the Earth Biogenome Project. I think you might be familiar about this multi-institutional collaborative project in which together with the top scientists and top institutions in all fields of life science around the world, we have collaborated with the core goal to sequence, catalog, and characterize the genome of all the Earth's eukaryotic biodiversity to understand the origin, evolution, and biodiversity of the eukaryotes. And if we talk about the research direction, we have three main research direction about the animal, plants and ocean. But in the today's talk, I will be mainly covering about our plant research. And overall, the aim of our team is to decipher the genome of all life on Earth and generate more than 1 exabyte of data and scientifically solve the key problems, especially of the species evolution, and to promote the ecological protection and utilization of these important biological resources. So far, our team has published more than 120 articles on animal and plant genomes, which accounts for more than 30% of the related articles worldwide. And in the subsequent slides, I will be talking about some of these important findings. Before moving forward, I would like to introduce briefly about the whole genome sequencing for the young audience. This is a technology which allows us to reveal the complete DNA, or we can say the genetic makeup of an organism, and it also allows us to better understand the variation both within and between the species, and it allows us to differentiate between the organism with a precision that others technology do not allow. Yes. So this has led to the emergence of agriculture genomics or agrigenomics, which is the application of genomics in agriculture to improve the production of crops and livestock. And over the past 3 decades, we have witnessed a massive improvement in the technologies, especially due to this improvement. Now, the cost of genome sequencing has dropped dramatically. If you remember, wonder, in 1997, when the first human genome was sequenced, it costed around 3 billion US dollar, and it took around 10 to 15 years to complete. But now with this advancement, we are now able to sequence a genome at less than 300 US dollar. We can, and people even planning to reduce further to $100, and the whole genome sequencing and assembly can be completed within hours instead of 10 to 15 years. So this is how the technology has improved, and now we are applying this, the latest technologies with reduced cost to solve and answer various important biological questions. Crop plant genomics is emerging as the agriculture for future. If you see globally, around 391,000 plant species have been identified, among which 5,530 species alone are used for food for a human throughout the history. But interestingly, only three crops, rice, maize, and wheat, currently provide more than 50% of the world's calorie. And after species selection and sequencing with the help of proper breeding technology followed by the industrial production, the number of crop species having more nutrition values can be increased. This is the general scenario these days being followed. And this genomics technology is now significantly being applied for various model crop plants. And if you see over the past 2 decades, we have sequenced majority of the commonly used vegetable and crops. And in the recent year, there has been a dramatic increase in the sequencing of the pan genome of the whole varieties to get more detailed insights to identify their unique QTLs, markers, and to improve the breeding strategies. If we look at the agrogenomics market and trend, it is estimated that the global genomics, agrigenomics market will be 5.3 billion US dollar by 2026, growing at the rate of 9.7% during this year. And this rapid advancement in the functional genomics particularly will definitely bolster the overall growth. And China and Australia exhibit the potential to grow at more than 7.3% over the coming years. And China alone will add approximately 1.2 billion in terms of addressable opportunity for picking up aspiring businesses and their smart leaders. And it is estimated that the genomics and marker assisted selection will be the fastest growing segment in the Asia Pacific region. 10 years ago, our BGI team, in collaboration with various scientists and institutions across the globe, launched this 1000 plant transcriptome project, in which our aim was to phylogenomics. Using phylogenomics, we want to understand the evolution of the plants. And in this study, we generated 1,345 transcriptome data from more than 1,174 plant species to gain a better understanding of the plant phylogenomics. But transcriptome data was not sufficient enough to answer all the scientific questions. Therefore, in 2017, we launched the 10,000 plant genome sequencing project with the aim to sequence the complete genomes of more than 10,000 plants and protists, which comprising almost 8,000 seed plant genera, 1,000 non-seed plant genera like ferns, moss, liverworts, and hornworts, and also around 1,000 green algae and other photosynthetic and heterotrophic protists. And this slide shows a general strategy, a workflow of our 10,000 genome sequencing project. After collecting the samples, we usually perform the DNA extraction, and before doing the genome sequencing, we first perform a genome summary to assess the genome size and other basic parameters. Then we perform the draft genome assembly using second and third generation sequencers. Different kind of strategies depend on the scientific questions and the genome in our hand, the plants in our hand. And for the selective species, we also perform the chromosome scale assembly using the Hi-C library sequencing data. So this is the general strategy we follow for most of the plants we sequence under this project. If we look at the current progress of our 10KP project, so far we have received more than 3,000 samples from different parts of the world, mainly from China, US, Germany, and among which we have sequenced about 1,700 species, and 437 species has been already assembled. And in this we have different research direction and different clades for different scientific questions. Yeah. Coming to the main agriculture topic, we have a collaboration with African Orphan Crop Consortium with World Agroforestry Center in Nairobi, Kenya. And in this project, we plan to sequence 101 African orphan crop species. Orphan crops are nothing but the neglected crops, which have very high potential to become the common crop. Like they have very high yield, they are highly climate resilient, they are also biotic, antibiotic tolerance, they have very good harvest and good nutrition, even the good market value. But they are not being cultivated or utilized at the large scale like other crop species, we can say rice, wheat, and maize. So, in order to understand their genetic mechanism, their genetic diversity, and why these plants are able to thrive and have very good resilient features to adapt to various climatic conditions. Understanding this important feature would enable us to further improve their breeding, to improve their variety, and ultimately improve the livelihood of the local people. And so far in this project, we have completed the genome survey of more than 20 species out of 101, and we have gained some important insights regarding their key evolutionary features. And here in the red color one shows the genomes we have sequenced and published. For example, Vigna species, Faderbia albida, and Sclerocaria beria, especially this Moringa oleifera, and the eggplant. And others are in already in the pipeline. And I would like to tell one example about the Atrocarpus plant, the jackfruit and breadfruit, which are the largest fruits in the world. And this genome help us to explain their energy dense fruit composition and also the underlying genes involved in the sugar and the starch metabolism particularly. And we identified a total of more than around 2,800 and 2,000 expanded gene families in jackfruit and breadfruit respectively. And when we further did the function annotation, we found that the copy number of these several starch synthesis related genes were found to be increased in both jackfruit and breadfruit compared to other closely related species. And we also performed the transcriptome analysis and found that the tissue specific expression might actually imply their sugar rich and starch rich characteristics. Yeah. And we hope that this genome annotation can be used to accelerate the genetic improvement of these crops. And using our genomic approaches in the past 10 years, we have, in collaboration with various institutes worldwide, we have developed the genomic resources for all, several important crops such as maize, pigeon pea, and this high oil crop sesame, pearl millet, chickpea, and recently also for several rice varieties, and many others are in pipeline. And in the subsequent slides I will be covering some other important crops which are not mentioned here. Now I will tell one case study using the lettuce genome. As you all know, lettuce is an important vegetable crop widely consumed as a salad greens in many countries. And modern lettuce varieties are classified based on their morphological characters into like coarse, butterhead, crisp, Latin, and cutting, and the non-leaf type like stalk and oilseed. And it has, as it suggests, they have various agronomic traits and they have well-preserved germ plants. And it's a model asteraceae plant. So as I just mentioned in the previous slide, lettuce are divided into various types based on leafy and non-leafy types. And these are the crisp, butterhead, coarse, stalk stem, and oilseed varieties. And Lactuca cereola is actually a proposed wild progenitor of the lettuce crop. So in this large scale study, we have used different varieties of these lettuce to decipher their evolution and origin. So using the lettuce genome, we try to solve some of the scientific question about domestication, like where was lettuce domesticated and when was lettuce domesticated? What are the key events during the lettuce domestication? And what are the genetic determinants for the domestication traits? And to resolve and to solve this question, we use 445 lines of from 47 different countries and representing mainly the four species of Lactuca. And here the color graph shows the sampling site across the world. As wild relative species represent an important source of genetic diversity, understanding their phylogenetic relationship with the cultivated lettuce is of great practical importance in lettuce breeding. And using the phylogeographic grouping, we identified 3 European groups and 3 Asian groups for the lettuce plant. We divided the Lactuca accession into 6 major groups according to their phylogeny and geography, mainly into WE to CAS, and to determine the relationship within the cultivated lettuce. We estimated the genetic differentiation from Lactuta sativa and the nucleotide diversity of these 6 phylogeographic groups of Lactuca seriola, and overall the CAU group, this group had the smallest FST value compared to the other cultivated lettuce and within each of the 4 crop types. So among all these, the CAU group accompanied the most abundant singleton as well and the highest linkage disequilibrium decay rate, indicating that this CAU group representing the Caucasus is the center of origin of Lactuta worldwide. As you can see in this slide very clearly here, the red colored dots, here we have found the most abundant singleton in the Caucasus, suggesting that this is the origin or the main domestication center of the cultivated lettuce. To be very specific, the Vavilo region is actually the center of origin. And next, we also studied the divergence of between L. sativa and Seriola. And as a little archaeological evidence has been discovered for lettuce so far, we try to estimate the domestication time by assessing the change in the effective population size between these 2 species, and our result shows that around like 10,000 years ago, both these species as having a very stronger contraction, and which continued for the next 2,000 years. But if you look at the divergence pattern for both the species, we found that both the species diverge around 6,000 years ago, which denotes the domestication of the cultivated lettuce. And all these results together confirm that lettuce was first domesticated near the Caucasus region, and this European L. cereula population played an important role in the crop improvement after the lettuce was spread to the mainland Europe. Next, in order to better understand the impact of human selection on the lettuce genome, we carried out genome-wide association study of the domestication traits in the cultivated lettuce. Some of the domestication traits such as seed shattering, leaf morphology, leaf spines were used, and we've identified several important QTLs or genes related to these morphological features. You can see the distinct peaks here representing the distinct morphological traits. Then next we focus on the origin of these important domestication traits like seed shattering, leaf spine and leaf morphology, and our analysis clearly showed that seed shattering was originated in the Iraq domestication and also the leaf spine. However, the leaf morphology was originated in the Italy during its domestication. So, if you look at the overall history, based on our overall finding, we propose the domestication history of the lettuce, and of course, CAU that else Lactuca seriola was the domestication site or the origin of the whole domestication pathway. Then it went to the ancient lettuce, then oilseed varieties. So here the green boxes are all the cultivated varieties, and in this the purple frame is the L variosa here, and cross actually represent the potential integration process. Yes, and the genes, for example, QLFD, QSHT, and this actually represent the three loci controlling the leaf morphology, seed shattering, and the leaf spine, which corroborates with our previous slide about their origin in Iraq and Italy. In the next section, this section actually, I am going to talk about the application of metagenomics in the plant root microbiome. As it is widely known that the plant lives with diverse microbes in nature. For each gram of soil, there are 10,000 different microbial species and even more. The soil microbiome represents one of the most complex microbial communities so far, and how they affect the plant growth, their nutrition uptake, and their disease response is now getting, the understanding is now getting better and better with the application of the metagenomics in this field. In general, there are two kinds of microbes. One is beneficial microbes and harmful microbes. And as we all know, harmful microbes cause different harms to the plant that cause several diseases, causing the significant loss in the crop yield or total death of the plant. However, on the other hand, the beneficial plant associated microbes have various impact on the plant phenotypic plasticity. For example, it enhances the plant nutrient acquisition. It increases the plant tolerance to abiotic stress. It promotes the plant growth and productivity. It also affects the plant secondary metabolite accumulation. And overall, it helps the plant to resist pathogens. Yes, so it's very important to identify the beneficial microbes to plant performance such as growth, yield, and quality, so on. And that could be ultimately applied for the agriculture practices. And if you look at the root associated microbiomes, it can be mainly divided into three parts. First, rhizosphere soil, that root associated microbe that could be divided, the first one here, it represents the microbiomes living in the soil zone with less than 1 millimeter distance from the root. Yes. And the second one is the rhizoplane microbe. It means the microbes lives on the root surface and can only be eluted by ultrasound. And the third one is the endophyte microbiome. It means the microbes living in the internal tissues of the plant. By using the two case study, one is citrus root microbiome, that is orange microbiome, and another one is the foxtail millet root microbiome. I am going to explain how we have applied this metagenomics to understand the plant root microbiome and their impact on the overall growth of the plant. The dried orange peel is a traditional herbal medicine in various countries, especially in China. And this medicinal citrus cultivar, particularly the orange species called Citrus reticulata chachi, is widely cultivated in the southern part of China, such as Guangdong and Guangxi province. And these two places are 283 kilometers far away, but the citrus quality in these two places significantly differ from the planting region. For example, the citrus peel from the core region, like Xinhui core region, is very high yielding and the quality is highly superior compared to the other places, particularly the essential oil, which is one of the major bioactive component in the citrus orange peel. So the question comes, how the environmental factor contribute to the qualitative difference between the core region and the non-core region. And this study, we mainly focus on the effect of the soil and the microbiome on the bioactive component essential oil, that is essential oil in this citrus peel, and what is their effect. So this was the main question we have used to address using this research. So in order to gain further deeper understanding, we performed a comparative analysis of the essential oil in the citrus peels collected from different planting region. And interestingly, we found that a total of 14 monoterpenes were detected in the citrus peel, and our comparative analysis showed that 7 out of 14 of these monoterpenoids were significantly abundant in the core region. For example, ocymene, beta myrcene, and alpha pinene, etc. And then we mainly focus on these 7 monoterpenoids, and we try to find the factors correlated with these monoterpenes, including the soil property and microbiome and the host gene expression. Yes. Next, to identify the association of the special soil property and the microbes to this newly identified 7 monoterpene abundant in the core region, we performed the correlation analysis, which showed that manganese, magnesium, potassium, and soil salinity were positively correlated with the monoterpene biosynthesis. And actually, manganese and the magnesium, they are very important cofactor of beta pinene synthase and 1-DXS in lemon. And several of these highly expressed genes in the core region were correlated with the monoterpenes, including the salt responsive genes like calmodulin, putative ATPase, ABA receptor, etc. In addition, we also identified several plant immunity related genes such as VORKI1, MKS1. They were highly positively related with the monoterpenoids. Our further analysis led us to gain more insight into the root microbiome, and we found that this root microbiome and soil nutrition actually boosts the accumulation of monoterpene. We found that 8 MAG genera significantly correlated with the content of monoterpene and were also correlated with the plant immune genes such as MKPK3, OX1, RPM, and so on. This indicates that microbes in the rhizosphere soil might contribute to the accumulation of monoterpenes by triggering the plant response. Therefore, we isolated one endophytic strain called Serratia marcescens and from the citrus root planted in the core region. And we also performed the genome sequencing of this species. And interestingly, this species showed that it has the complete terpenoid backbone pathway, and therefore we hypothesize that this might synthesize the intermediate product of the monoterpene for the plant, for the citrus plant. And in summary of this citrus research, we could say that a special soil environment like rich in high salinity, magnesium, manganese, and potassium actually enhances the monoterpenoid content by promoting the expression of the salt stress responsive genes and and also the activity of the terpenoid backbone synthase. And the rhizosphere soil, on the other hand, along with the endophytic bacteria, can co-contribute to the monoterpene accumulation by triggering the plant immune response or by providing the intermediate products for the plant. Our next case study is on foxtail millet, and this is another very commercially important agriculture crop. And our genome-wide association study provided very interesting insight into the precision genotype dependent microbial effects in foxtail millet. Here, by using different metagenomic approaches, we try to answer 3 main questions like how the host genotype and root microbiome can contribute to the plant phenotype? And what are the potential mechanism of the host genotype interaction with the root microbiome? And whether there is a genotype dependent microbial effect on the phenotype of foxtail millet. So we use the GWAS, MWAS, and MGWAS method to analyze the association between the genotypic, phenotypic, and root microbiome from 827 cultivars. And these cultivars were collected from different parts of China. At first, we performed GWAS on the phenotype and identified 86 significant SNP loci and 91 association for the 10 various traits. And the candidate genes around the SNP loci involved in both abiotic and biotic stress response actually implied that the host genotype and the environment interaction might contribute to the growth and yield of the foxtail millet. In the next step, in order to validate the effect of the marker OTUs, we isolated the bacterial strains from the field and 10 strains with the top beta estimation in the regression model we selected for the further validation experiment and found that 4 bacteria particularly showed very good growth promoting effects in the foxtail millet and 2 bacteria showed growth suppressing effect on the foxtail millet. Further, we ran MGWAS on 1004 common OTUs to identify the association between the genetic SNPs and microbial OTUs. And interestingly, we found that 838 microbial OTUs with significant SNP loci. And the function of the host genes around the significant loci mainly were related to immunity, metabolites, hormone signaling, and nutrient uptake. This clearly indicate that the potential mechanism of the host plant for regulating the microbial structure. We can also see that the abundance of the marker OTUs shifted among the different genotypes at the most strongly associated SNPs. For example, this one, SUZ12. This is a candidate gene which is associated with the abundance of the marker OTU 19835, and the abundance of this was higher in the allele genotype cultivar, while but lower in the reference genotype. So this means that there might be a different effect on the phenotypes when we inoculate the marker strain with a different genotype cultivars. So in summary about this project, we examine the association between the genotype, phenotype, and the environmental variable from 827 foxtail millet cultivars by using GWAS, MWAS association studies, and particularly our MGWAS study revealed that the root microbiota composition was mainly driven by the variation in plant genes, particularly related to the immunity, metabolites, hormone signaling, and nutrient uptake. And strikingly, we found that the root microbiome affect the agronomic phenotypes in a host genotype dependent manner. And if we talk about the future perspective in the plant microbiome research, it is anticipated that MWAS will facilitate the identification of the microbial markers for the plant health, growth, and overall productivity. Especially the beneficial microbes is useful for agriculture practice, and which actually will be reducing the chemical fertilizer, our less reliance on them. it will improve the soil quality, and so on. Thus, it is very important to isolate and identify the strains which will affect in the field experiments in the future. And MGVAS could help us to reveal the association of genetic variation of root and especially the root microbiome. And in future, the usage of multi-omics like metabolomics, transcriptomic method can be applied to decipher the potential mechanism of plant and the microbe interactions. And genome editing could significantly speed up the progress of plant breeding. Yes. And genome editing technologies consisting of targeted mutagenesis and gene targeting now enable us to modify genes of interest rapidly and precisely. Its discovery in 2012, the CRISPR-Cas9 system, and the subsequent development in the sequence-specific nucleases has brought a paradigm shift in biology. And later, with the Nobel Prize for the CRISPR-Cas9, now it is continuously being applied for various application in agriculture or plant improvement, we can say. For example, the application of genome editing to crop breeding has particularly advanced in parallel with the development of the genome editing technology specifically. And at present, I can say it is no exaggeration to say that we are now in a position to be able to modify a gene precisely and rearrange genome and chromosome in a predicted way. And therefore, now the genome editing application has a various application, for example, in biotic stress response, abiotic stress response such as drought, salt, heat and cold, and to improve the nutrition. So, for example, the oil production, phytic acid, starch, and vitamins, herbicide resistance to make control the weed, uncontrolled growth of weed can be controlled using this, and to mainly to improve the overall yield, like for example, grain weight, the grain size, and the grain number. So ultimately, this is the future. And I would like to add one more thing that in today's world, almost like 1 billion people are now suffering from chronic malnutrition. And while at the same time, our agriculture system are degrading, we are suffering exacerbating loss of biodiversity and with increasing uncertainty, especially in the climate change. And with the global population projected to exceed 9 billion by 2050, this contemporary agriculture will face enormous challenge, and it will definitely require crops with higher yield and improved quality and needing fewer inputs. And although conventional breeding is currently the most widely used approach in crop improvement, it is quite labor intensive, and it is usually takes several years to progress from the early stages of increasing screening the phenotypes and the genotypes to the first crosses into commercial varieties. Therefore, I strongly believe the application of omics, genomics, and the genome editing will definitely improve the overall, or it will definitely change the overall agriculture field. And with this, I would like to share about our 10,000 plant genome progress, the website, and how the whole workflow actually works. And if you're interested, you are most welcome to visit our website. Here we have the online submission, batch submission detail, and we have our strategy timeline, what are the species we have sequence, what are our priority list, everything you can find all the detail. And if you see the brief summary of our workflow again, so since it is a community effort, we have the samples coming not only from China but across the globe, And first we do the nucleic acid extraction, the RNA and high molecular weight DNA extraction, followed by the library preparation and sequencing. And depending on the target species, we select the genome sequencing method, and using our in-house assembly annotation pipelines, we perform the data analysis. And once the data is generated, we usually build various databases. And most important thing here is all the data, all the genome, all the metagenome data we generate in this project are released publicly and are freely downloadable using our China National Gene Bank database, also our partner GigaScience Biodiversity Data Release database. So this is an ongoing, continuous project, and if you are also having some interesting plant species or interesting project ideas, you are very much welcome to directly contact us so we can discuss further. Since 10,000 plant genome project is a huge project, of course we have several challenges. For example, first we need to improve the overall workflow, need to be more transparent, need to be process need to be established, and especially for our collaborators, we need to establish some customer feedback process. And one of the major challenge here in this project is to sample shipment. Especially our collaborators usually collect samples from a remote island or remote forest, and by the time they collect the sample and ship to China, most of the time the samples are degraded. And if they send an extracted DNA or RNA sample, majority of the time when we receive the sample, it's degraded. So this process we are also trying to overcome by establishing or by troubleshooting or by maintaining, trying different protocols and optimize extraction methods for this. Similarly, we also need a more efficient way of data information sharing, because the amount of data generated using for this large scale genome sequencing project, it's usually in the GB, several gigabase of data are there. So data sharing is also sometimes a little bit problem. And regarding the technical improvement, we are trying to develop more time and cost effective sequencing strategy. For example, BGI has developed one single tube long fragment reading, that is, now we can generate long read sequencing using the second generation sequencing data. Yes, and so that makes the whole process quite cheaper and very effective for the sequencing strategy. Yes, and of course, due to the large number of data, we also need to improve and develop more better assembly pipelines for more faster and more better genome assemblies. Of course, then sample preparation, especially for the high molecular DNA extraction, need to be further improved, especially for the plant sample, where we receive a wide variety of diverse plant species. Sometimes the same protocol is applicable or suitable for one plant species but doesn't work for other. So this need a serious optimization. And last but not least, due to continuous improvement in the sequencing technology, of course, the cost is also increasing actually for this. So therefore, we need more funding for this project eventually. And finally, I would like to acknowledge our 10,000 plant genome working group from different institutions worldwide, for Professor Gain Shu Hong, Professor Soltis, Doug Soltis, Pamela Soltis, Professor Michael Melkonian, Yves Van de Peer, Sean Graham, Shushan, our BGI group leader, and Stephen A. Smith, and our supervisors, Liu Xin and Huan Liu, for their constant support and encouragement. And finally, I would like to thank you all for your patience listening. And if you have any questions, you feel free to reach me via my email or mobile. And if you're in China, if you use WeChat, you can directly contact me through that also. Yeah. Thank you. I will be very happy to take your questions. And once again, I thank the organizer for giving me this excellent opportunity to share interesting findings in front of you all. Thank you. Thank you so much.